Introducing Ingenuity variant analysis, a knowledge-driven way to find causal variants in next-generation sequencing data fast. DNA resequencing experiments yield millions of variants per genome. Identifying the ones that impact disease progression or phenotypic differences can be as difficult as finding a needle in a haystack. How can you quickly and confidently find the variants that are worth following up on? Ingenuity variant analysis is the fastest way to identify causal variants by providing a single integrated source for both genetic and biological analysis. It quickly identifies scientifically compelling variants for you. This allows you to focus on the biological significance of your data. Let's have a look at the features of Ingenuity variant analysis that can help you do this. Imagine you are investigating the causes of a hereditary disease by comparing the genomes of two affected children with those of their unaffected parents. First, you or your sequencing service provider would securely transfer the variant call data generated from each of the biological samples into Ingenuity variant analysis. Next, you would create an analysis by simply choosing a study type, specifying relevant phenotypic terms, and selecting the sets of samples to compare. After opening the analysis, you see a short list of phenotype-relevant variants to review that are the result of applying a series of filters based upon your personalized analysis settings. The Common Variance filter excludes variants that have a population frequency greater than your disease prevalence. The Predicted Deleterious filter selects for variants that are either known to have a phenotypic effect based on the Ingenuity knowledge base, or are inferred to cause a gain or loss of function. The Genetic Analysis filter interprets genotypes and copy number variation for each of the samples in your analysis to identify variants among them that are specifically associated with the phenotype. Filters are easily adjustable and others can be added, dynamically updating your results. The Biological Context filter enables you to interactively ask biological questions of your data. Ingenuity variant analysis provides accurate answers by leveraging the Ingenuity knowledge base, which consists of millions of biological and pharmacogenetic findings from the peer-reviewed literature and commonly used databases, describing relationships between variants, genes, pathways, processes, drugs, diseases, and more. These relationships are individually modeled and manually reviewed for accuracy, and they always include a link to the source so you can review the evidence supporting each relationship. Simply enter biomedical terms from any scale of biology, from protein domains to disease symptoms, and this filter will translate them into the corresponding established variants and genes from the Ingenuity knowledge base. To find novel causal variants, you can identify variants in your data set that impact genes upstream of the established disease-implicated genes. Furthermore, you can apply causal reasoning to select for variants that are causally consistent with respect to their molecular effect on known disease genes. The genotype and inferred functional impact of each variant is visualized for each sample. In this example, the two affected children both have a homozygous loss of function for a variant which is heterozygous in both parents. Clicking on a row in the variant table provides a detailed view of the variant by sample and across transcripts of the gene. It also provides a means to see the literature-supported path from your variant to the disease. Clicking on a line connecting two genes displays the findings from the Ingenuity knowledge base that support the directional molecular effect. In this example, you've now discovered a causally consistent disease variant for further follow-up. Ingenuity variant analysis also provides a data-driven approach to identify variants within genes, pathways, biological processes, and diseases that the genes containing your deleterious variants are implicated in. You can use this information to focus in on variants with phenotypes relevant to disease biology. Ingenuity variant analysis is an interactive, accurate, knowledge-driven application that enables you to quickly identify causal variants in your human resequencing data. There's no other tool like it.